Dalston Park is a 320-acre national nature reserve stretching along the coast of the Isle of Purbeck in Dorset, England. Due to the area having dramatic sea cliffs with large elevation due to mining having taken place there, it is a great area to investigate how sodium chloride, salt, affects the different types of plant species moving away from the coast. We should expect to see plants such as kidney vetch and thrift closest to the coast, and great burnet with bird foot truffle at the furthest transect from the cliff. The difference in species depends on their tolerance to sodium chloride, as salt dehydrates the plant, and if it is not adapted to dealing with the conditions, it will wilt and die. This occurs because plant cells attract water by having a higher concentration of salt or other dissolved compounds inside the cell than outside the cell, so water moves across the cell membrane, known as osmosis. However, if there is more cell outside of the cell than inside, then the water moves from inside the cell across the cell membrane to outside of the cell, causing plasmolysis and wilting. A secondary factor we looked at is altitude and how that may affect different species of plant occurring in each area, starting from the lowest altitude at the lighthouse. Because the altitude will not be measured at set intervals, a cause and effect cannot be drawn, but a relationship may be seen. Furthermore, other aspects to consider would be how trampling, specifically by humans, have an effect on plant species, by observing the richness of species over paths, and also how sheltered the area is by hills and large shrubs. Dalston Park is a
Right, so the method. Quickly going through the equipment, we had a 1 by 1 meter quadrat, a 5 meter tape measure, and then just the notebook and pen to write stuff. Our general idea, with the aim in mind, we just sort of tried to cover as large of an area as possible, obviously considering the time frame we were given. This would obviously allow us to look at a large contrast of areas, and hopefully get a large contrast of species, since we're looking at a larger area. Marking out the sites, we used our tape measure to make a 10 by 10 meter square. We just marked each corner of the square with like our backpacks. And this 10 by 10 meter square was basically our designated site. We used 6 different sites in total, and each one was relatively random. The one constant thing was every 2 sites we moved further inland. Our quadrat, we had a 1 by 1 meter quadrat, as I said before. In each site, so each 10 by 10 meter square, we threw the quadrat 10 times. And then each species in the quadrat we recorded. The species were just marked A to J, mainly because we didn't know the name of every species. And each time one of these species was seen in the quadrat, it was recorded. And at the end of each site, we tallied up the number of plant species in that site. As I mentioned before, it was fairly random, the location of the different sites. We tried to have 2 sites the same distance from the coastline, mainly to see if each species was constant and if any other factors played a part, such as elevation, soil type, stuff like that. Sites were quite a large distance away from each other. This was, again, just to cover a larger area. So site 1 was as close to the coast as possible. We worked on the site the day before, so it was a good place to start. Site 2 was about 100 meters southwest of site 1. It was roughly the same distance from the coast. Then we moved further inland, about 250 meters northwest. Site 4 was about 200 meters from that site. Again, same distance from the coast as Site 3. This one, other than Site 1, was the only site we sort of purposely chose. Since it was on top of a hill, we wanted to see if altitude sort of played any part in the species that were there. And then Site 5 was quite a bit further inland, again to get a nice contrast to species, hopefully. And then Site 6 was just a little bit further east from Site 5. So yeah, thank you very much. That's the method.
Starting with graph 1, you can see that data from areas 1, 2 and 3 have been used. Species is marked along the x-axis, ranging from A to J, and abundance is marked along the y-axis. Abundance has been calculated by the total number of times it was seen in 10 quadrats. We can see that G, H, I and J have only been seen in areas 3. Graph 2 follows the same format using data from 4, 5 and 6. We can see that species A, E and F have not been found in these areas. Finally, we can see this line chart is marked with abundance along the y-axis again, but this time the x-axis follows areas 1, 2, 3, 4, 5 and 6. The blue line marks out species A. Species A is found in areas 1 and 2, and it is not found again. Species J was not found in areas 1 or 2, but has been seen in areas 3, 4, 5 and 6.
Delston is a site of high species diversity and high species abundance. Part of the investigation that was completed was to identify 10 species that were picked up throughout Delston Country Park and to see which were more salt tolerant than the others. Therefore, this is to see if Delston has happened to halophyte species. When completing the investigation, factors that were considered as well as salt tolerance were if there are past the investigation sites that indicate trampling, if there are protection and shelter for plants to grow in, and also to see if plants were being out-competed by other tall vegetation that would have more access to sunlight and nutrients in the soil. Bias was tried to be eliminated by doing random drop sampling, quadrat sampling, using a dice online, and the reliability of the results collected was increased by doing 10 quadrats at each transect site, therefore doing 60 quadrats in total. The kidney veg was seen to be a salt-tolerant plant as it grew very close to the coastal edge and not far away. Transect line that we completed could be considered a bit biased as it was not particularly straight inland, not a belt transect, it was a bit zigzagged, and therefore we could not go as far inland as we wanted. It was also hindered by fields that you could not trespass in. The first species that we identified, species A, is a salt-tolerant plant as it grew very close to the coastal wedge and was only found up until about a couple hundred meters inland, whereas the Great Burnet species, species J, it seems to be less salt-tolerant as it started growing a couple hundred meters inland and then grew in sites 4, 5 and 6. As in terms of linking our investigation to the wider world of science, people could complete more investigations on the kidney vetch along different coastal regions in the UK to see if there are similar trends for the kidney vetch elsewhere. More investigations could also be completed at Delston Country Park, looking at the 10 species that we picked up and using different sites along the coastal edge and inland, and compare the data collected there to the collected data in our investigation.
The aim of our investigation was to compare the abundance of plant species from the coast inland. We had 6 sites, 1, 2 and 3 which were near the coast, and 4, 5 and 6 which were further inland. We found species such as thrift, scurvygrass and kidney vetch only in sites 1, 2 and 3, while species such as great baronet and bulbous buttercup only in site 4, 5 and 6. This is because the plants found only in sites 1, 2 and 3 are haliophytes, which means they're they can handle salt tolerant, whilst species such as bulbous buttercups and great baronet are less tough and can't handle the salt. What we can learn from this is that if you were to study for specific plants, it is really important to know what their conditions they live in. Is such as kidney vetch is vital to a butterfly species called small blue, which is endangered. And if you were studying the butterfly or all the plant, and you were to try and find them, you could aimlessly walk around a field and you'd never find one, or you could aim straight for the coast and probably stumble upon one really quick. And the same goes to bulbous buttercup, like you wouldn't find any at the sea, so there's no point wasting your time around there. So the general aim that we learned was that within a couple hundred meters from the coast inland, plant species can be completely different. And so if you're studying, it's really important to know the species you're after and the conditions, because a couple hundred meters can make that big of a difference.